If you have a broad nose, wide nostrils, a flatter bridge across the middle of your face, somebody has probably commented on it. Maybe a stranger, maybe a relative at a family dinner who thought they were being funny, maybe a mirror on a bad day when you were looking for something to criticize and it was the easiest target in the room. What almost none of them knew, and what you probably don't know either, is that the shape sitting on your face right now is the physical signature of a continent that no longer exists, a population that survived there for 50,000 years, and at least one interbreeding event between our species and another kind of human that vanished from the Earth before recorded history began. This isn't metaphor. This isn't the kind of soft, feel-good framing people use to make you feel better about a feature you didn't choose, the sort of thing you might see printed on a motivational poster and immediately dismiss as empty comfort. This is published, peer-reviewed, genome-sequenced fact, sitting in academic journals that almost nobody outside a small circle of population geneticists ever reads, buried in dense statistical language and technical figures that were never written with an ordinary audience in mind. And once you actually understand where that trait comes from, once you trace it back through the real research instead of the simplified version that gets passed around online, it becomes very hard to look at it the same way again. Let's start with the study everyone thinks they know, because there's a widely repeated claim floating around that the genetics of nose shape were finally cracked in 2023. That date is wrong, and the correction actually matters, because the real paper is nearly a decade older and far more specific than the popular version gives it credit for. The actual breakthrough came in 2016, published in the journal Nature Communications, led by a team including geneticist Kaustuv Adhikari, and it pulled genome data from more than 6,000 participants, recruited primarily across 5 Latin American countries, through a research effort called the Candela Consortium, a population chosen specifically because of its extraordinary genetic diversity. Earlier research into facial genetics had almost entirely focused on European populations, which meant an enormous amount of human variation was simply invisible to science, buried under sample sets that never included the ancestral mixtures found in places like Brazil, Colombia, Peru, Chile, and Mexico. This study fixed that gap, and what it found reshaped how we understand one of the most visible traits on the human body. Because, for the first time, researchers had enough genetic diversity in a single dataset to actually see patterns that a European-only sample would have completely missed. Instead of treating nose shape as one broad, blurry category, the researchers broke it down into distinct measurable traits: nostril breadth, nose bridge width, the angle and protrusion of the tip, nose length. Each of these turned out to be controlled by different genes, acting somewhat independently of one another, which is part of why no two faces ever look quite the same, even within the same family. 4 genes stood out with the strongest statistical signals: DCHS2, which showed a clear association with how pointed or blunt the tip of the nose is; RUNX2, tied to the width of the nasal bridge; GLI3, which produced the single strongest signal of all for nostril breadth; and PAX1, which also contributed to nostril width. A 5th gene, EDER, turned out to influence chin protrusion rather than the nose itself, but it showed up in the same dataset and became part of the broader story of how facial architecture gets built at the genetic level. Here's where it gets interesting, and where most coverage of this study stops short. DCHS2 is a cell adhesion gene involved in cartilage development, part of a regulatory network that shapes how the nose forms structurally during fetal growth. RUNX2 is a bone development gene, one that's been studied for decades in an entirely different medical context, because rare mutations in it cause a serious skeletal disorder affecting how the skull and collarbones form. Nobody had connected it to ordinary nose width variation until this study mapped it directly onto that trait. GLI3, meanwhile, sits inside a signaling pathway responsible for some of the most fundamental patterning decisions an embryo makes, deciding not just how wide the nostrils will be, but influencing limb and organ development elsewhere in the body. These are not throwaway genes. They are core architectural instructions, repurposed by evolution to produce the specific facial variation we see walking around today. It's worth sitting with that for a second, because it means nose shape isn't controlled by some minor cosmetic switch tucked away in an unimportant corner of the genome. It's built out of the same fundamental machinery that constructs the skeleton, regulates cartilage formation throughout the entire body, and coordinates how an embryo's face assembles itself in the earliest weeks of development. Later analysis of these same gene regions found something else worth noting: that DCHS2, GLI3, and RUNX2 all show strong signatures of recent evolutionary selection when compared against archaic humans like Neanderthals and Denisovans, meaning these particular genes were actively being reshaped by natural selection relatively recently in our evolutionary history, right around the time modern humans were spreading into new environments across the planet. adapting on the fly to conditions their ancestors back in Africa had never encountered before. And when researchers looked at how these gene variants are distributed across global populations, a clear geographic pattern emerged. Variants linked to broader, wider nasal features were found at much higher frequencies in populations with ancestry from Africa, Southeast Asia, and Oceania. Variants linked to narrower, more projected nose shapes clustered more heavily in European and parts of Northeast Asian populations. On its own, that lines up neatly with the textbook explanation for why nose shape varies around the world in the first place, something called climate adaptation. The logic behind it is simple and genuinely well supported by decades of anatomical research. In hot, humid climates, a wider nose with larger nostrils allows air to move through more efficiently without needing to be warmed or humidified as much, which is exactly what you want when the surrounding air is already warm. In cold, dry climates, the opposite holds true. A narrower, longer nasal passage warms and adds moisture to incoming air before it reaches the lungs, protecting delicate lung tissue from cold, dry damage. This correlation is strong enough that forensic anthropologists have used nasal architecture for decades as one piece of evidence when estimating a person's likely regional ancestry from skeletal remains alone. This isn't a new idea either. Anatomists were documenting the statistical relationship between climate and nasal cavity dimensions as far back as the 1970s, measuring skulls from populations around the world and consistently finding the same pattern: narrower passages correlating with colder, drier climates, and wider passages correlating with hotter, more humid ones. Decades of that kind of measurement gave climate adaptation a very solid evidentiary foundation, which is exactly why it became the default explanation taught in anthropology courses for the better part of half a century. But climate adaptation, as solid as it is, does not explain everything the genetic data shows. And this is the part of the story almost nobody tells you, because it doesn't fit neatly into a headline. Indigenous populations living at high altitude in the cold, thin air of the Andes Mountains carry these same broad nasal variants at frequencies that climate adaptation alone cannot account for. So do certain East Asian populations living in temperate regions, far from anything resembling a tropical climate. If environment alone were driving this trait, those populations should look genetically more like their cold weather neighbors. They don't. Something else was clearly happening underneath the surface. Something that pure climate selection over tens of thousands of years wasn't fully capturing. And the answer to that anomaly didn't come from nose research at all. It came from an entirely different, rapidly advancing field: ancient DNA. Over roughly the last 15 years, the ability to extract and sequence usable genetic material from human remains that are tens of thousands of years old has completely transformed what we know about human prehistory. And nowhere has that transformation been more dramatic than in Southeast Asia. For most of the 20th century, the accepted model of the region's history was relatively simple. An early population, sometimes linked to the ancestors of today's Aboriginal Australians and Papuans, occupied the region first. Then, somewhere around 4,000 to 5,000 years ago, a large wave of Neolithic farmers migrated south out of what is now southern China and Taiwan, bringing agriculture, new languages, and new technology, largely replacing or absorbing the people who came before them. That framework explained a lot, including the spread of Austronesian languages across an enormous stretch of the planet, from Madagascar in the west to Easter Island in the east. It was tidy. It was also, as it turns out, incomplete. The people who occupied the region before that Neolithic wave are sometimes linked archaeologically to what researchers call Hobbinian hunter-gatherer culture, a tool-making tradition found across mainland Southeast Asia that persisted for thousands of years before agriculture ever arrived. For decades, that culture was treated as a footnote, a transitional phase before the real history of the region began with farming. Ancient DNA has since made clear that dismissal was a mistake, and that the people behind it represented a far deeper, more genetically distinct lineage than anyone had given them credit for. When researchers finally sequenced ancient genomes from sites across Vietnam, Laos, and Malaysia dating back 8,000 years or more, the results didn't match either half of the old model. These weren't simply the ancestors of today's Aboriginal Australians, and they weren't simply early versions of modern Han Chinese or Taiwanese populations either. They were something distinct, carrying a genetic signature that included high frequencies of the broad nasal variants, one that didn't cleanly fit into any previously defined category. Researchers studying this population, sometimes described in the scientific literature as the first Sundaland people, trace their identity to a landmass called Sundaland, the exposed continental shelf that connected mainland Southeast Asia to the islands of what are now Indonesia, Malaysia, and the Philippines during the last ice age, when global sea levels sat far lower than they do today. At its largest, during the peak of the last glacial period roughly 20,000 years ago, Sunderland was not a narrow land bridge. It was a full continent, larger than Western Europe. with rivers, forests, and grasslands capable of sustaining a substantial, genetically distinct human population for tens of thousands of years. Genetic studies of the indigenous groups, sometimes referred to as Negrito populations, found today in the Philippines, peninsular Malaysia, and the Andaman Islands, show they diverged from West Eurasian populations at least 38,000 years ago and sit in a basal position relative to other East and Southeast Asian groups, meaning their lineage branched off earlier and more independently than almost anyone else's in the region. When the later wave of Neolithic farmers arrived from the north, they did not simply erase what came before them, they mixed, and the degree of mixing varied enormously depending on geography. On the mainland, blending was extensive, which is why populations across Vietnam, Thailand, Cambodia, and Laos today carry both ancestral lineages in roughly comparable proportions. In the islands, the picture is even more layered, with the Philippines in particular showing evidence of several distinct ancestral waves stacked on top of each other over thousands of years. But the broad nasal variants persisted through every one of those layers, every mixing event, every migration, because they were already deeply embedded in the Sundaland population long before any of those later movements ever happened. That still leaves one more piece of the puzzle unexplained, the part connecting Southeast Asia to the high Andes on the opposite side of the planet. Before the Sundaland population, before the Neolithic farming migrations, there existed an even older ancestral group sometimes referred to in the scientific literature as the Deep East Asian lineage, the shared ancestral population behind both the people who eventually settled Sundaland and the people who moved north and eventually became the ancestors of Northeast Asian populations. That same lineage is also the ancestral source for the small founding population that crossed from Asia into the Americas somewhere between 15 and 20,000 years ago, carrying broad nasal gene variants with them as part of their existing genetic inheritance, not as a fresh adaptation to any new environment they encountered afterward. That's the answer to the Andean anomaly. Indigenous populations across the high Andes, the Great Plains, and the Amazon rainforest carry these variants, not because their local cold or high-altitude environment selected for them independently, but because they inherited them intact from ancestors who carried that same genetic signature across a land bridge tens of thousands of years earlier, long before any of those specific environments existed as a selective pressure at all. It's a genuinely counterintuitive finding because the climate adaptation model would predict the opposite outcome entirely for a population that has lived at extreme altitude in cold, thin air for thousands of generations. But Ancestry, once it's locked into a founding population that's small, can persist stubbornly through environments it was never originally shaped by, simply because there wasn't enough time or enough new genetic variation entering the population for local selection to fully overwrite what those founders carried with them. This has real, practical consequences for anyone who has ever taken a commercial ancestry test and felt like their results didn't quite capture the full picture. The reference populations that companies use to calibrate their ancestry breakdowns are built almost entirely from contemporary genetic samples, which means they were never designed to account for 50,000 years of Sundaland population history or 20,000 years of subsequent genetic drift across the Americas. The practical result is that broad nasal variants in people with Filipino, Indonesian, Pacific Islander, or indigenous American ancestry frequently get underrepresented or misread in standard ancestry reports, not because the underlying genetics aren't there, but because the comparison data simply isn't deep enough yet to interpret them accurately. Researchers at institutions including Harvard's ancient DNA labs and large-scale projects like Genome Asia have spent the last several years specifically trying to close that gap by adding deeper ancient genomes and long underrepresented modern populations into their reference datasets. And what they keep finding is the same thing. The genetic complexity of Southeast Asian and Pacific Islander ancestry is significantly greater than what a simplified consumer report can currently show you. The Philippines is a particularly striking example of this. Genetic surveys of the archipelago have identified evidence of at least 3 or 4 distinct ancestral waves, layered on top of each other over thousands of years, each one contributing different genetic material to different regions and different communities across the islands. A single consumer ancestry category like Filipino is, genetically speaking, doing an enormous amount of compression, flattening thousands of years of layered migration, and mixing into a single word on a results page. None of that is a flaw in the person taking the test. It's a limitation of how young this particular branch of science still is, and how much catching up the underlying comparison data still has to do. Now for the part of this story that genuinely does involve a different species of human. Because that piece is real, it just isn't quite the version that sometimes circulates online. Around 60 to 70,000 years ago, when modern humans first began moving out of Africa, they encountered and interbred with Neanderthals. A legacy that shows up today as roughly 1 to 4% Neanderthal ancestry in the genome of nearly every non-African person alive. But the more striking archaic story for this part of the world involves a different group entirely. The Denisovans, identified from nothing more than a fingerbone fragment, a few teeth, and a partial jaw, first discovered in a Siberian cave in 2010. Despite that almost non-existent fossil record, researchers managed to extract a remarkably complete genome from those remains, and when they compared it against modern populations, they found something striking. This was, at the time, a genuine shock to the field. Nobody had predicted that a previously unknown branch of the human family tree was sitting undiscovered in a single Siberian cave, identified not from a skull or a skeleton, but from a fragment small enough to fit on a fingertip. A follow-up study a few years later, surveying archaic genetic signatures across more than 1,500 people worldwide, confirmed that while Neanderthal ancestry shows up broadly across nearly every non-African population on the planet, meaningful Denisovan ancestry does not. It's essentially absent outside a specific cluster of populations, which immediately told researchers that whatever interbreeding happened between modern humans and Denisovans, it happened in a specific place, at a specific time, involving a specific ancestral population, rather than being spread evenly the way Neanderthal ancestry was. Denisovan ancestry isn't spread evenly across humanity. It's heavily concentrated in specific populations, most notably Melanesians, with people from Papua New Guinea and the surrounding islands carrying somewhere between 4 and 6% Denisovan-derived DNA, a proportion notably higher than the Neanderthal contribution found across most of Europe and Asia. More recent research has pushed that picture even further, finding that the Aita people of the Philippines carry roughly 30 to 40% more Denisovan ancestry than Papuans do, putting their total close to 5% of their entire genome, the highest concentration of Denisovan DNA documented in any living population on Earth. What researchers have confirmed those Denisovan genetic fragments actually do to modern facial structure is genuinely remarkable, even if it isn't precisely the version of the story that gets repeated casually. A large, genome-wide study of Latin American individuals with partial indigenous American ancestry identified a specific genomic region tied to lip thickness that traces directly back to Denisovan introgression, one of the clearest documented examples anywhere in human genetics of an extinct human species directly shaping a visible facial feature in people alive today. Around the same time, a separate discovery found that attractive Neanderthal-derived DNA increases nasal height, the vertical length of the midface, a detail that lines up with what we know about Neanderthal anatomy, since Neanderthals themselves had notably tall, forward-projecting midfaces compared to modern humans. Taken together, these findings represent 2 of the only confirmed cases where archaic human DNA has been shown to directly influence the shape of a living person's face, and they establish something the broader nasal genetics research strongly suggests as well, that some of the facial variation carried by populations with deep Southeast Asian and Pacific ancestry, including the broad nasal architecture at the center of this story, sits on a genetic foundation shaped in part by interbreeding with a species of human that no longer exists, layered on top of tens of thousands of years of Sundaland ancestry and climate-driven selection. Researchers expect this list to grow. Archaic genomes are difficult to find and even harder to sequence cleanly, which means the 2 confirmed cases we have today almost certainly represent a small fraction of the full picture still waiting to be uncovered. There's also an open question in this field that honest researchers don't pretend to have fully settled, and that's the role of sexual selection, the evolutionary process by which mate preference itself shapes which physical traits become more or less common over generations. Some scientists think it played only a minor role in nose shape specifically, with climate adaptation and ancestral inheritance doing most of the explanatory work. Others argue that in smaller, more isolated ancestral populations, where individual mate choices can shift trait frequencies relatively quickly, sexual selection may have amplified differences that started out as climate-driven adaptations in the first place. Nobody currently has a definitive answer. And that uncertainty is worth stating plainly rather than glossing over, because it would be easy to build a tidy, satisfying narrative out of climate adaptation alone and simply leave it there. Good science resists that temptation. The honest position is that multiple forces were almost certainly operating at once, reinforcing each other rather than competing in some clean, isolated way, and untangling exactly how much weight each one carried is the kind of question that may never be fully resolved with the tools we currently have. By the time these populations show up clearly in the ancient DNA record, the broad nasal variants are already present at high frequency. Now bring this back to something personal, because underneath all the genomic terminology, that's what this research actually is. If you carry these variants, you are carrying genetic material passed down from a population that survived on a continent that has since vanished beneath the ocean. a population that interbred with at least one now extinct branch of humanity, and whose descendants then spread across a geographic range wide enough to eventually cover nearly half the globe, from the Philippines to Melanesia to the high mountains of South America. That is not a small inheritance. And for much of the 20th century, broad nasal features specifically carried an unfair cultural weight in Western beauty standards, sometimes treated as markers of difference rather than what they actually are, the visible result of tens of thousands of years of successful adaptation and survival. It shows up in real-world numbers, too. Cosmetic rhinoplasty rates in several East and Southeast Asian countries, including South Korea, Japan, and the Philippines, are among the highest anywhere in the world. And the most commonly requested changes specifically involve narrowing the bridge and reducing nostril width. The exact features this research traces back tens of thousands of years through ancestral inheritance. That pressure didn't come from nowhere. It came from decades of media, beauty standards, and casual commentary that treated one narrow version of facial structure as the default and everything else as a deviation from it, something to be corrected rather than simply understood. None of this research is meant to tell anyone what to do with their own face. Personal choices about appearance are exactly that, personal, and nobody outside your own life gets a vote in them. But understanding where a trait actually comes from changes how it sits in your mind. It's a lot harder to see something as a flaw once you understand what it took to survive 50,000 years, a lost continent, and interbreeding with a species of human that no longer walks this earth, in order for it to end up on your face today. The research itself is still moving, and moving fast. New ancient genomes get sequenced every year. The reference panels used by commercial ancestry companies keep expanding to include deeper, more geographically representative data. Researchers studying Denisovan introgression suspect there may be additional archaic populations that contributed DNA to Southeast Asian and Pacific genomes that haven't been fully characterized yet, meaning the picture painted here, as detailed as it already is very likely incomplete. 5 years from now, the ancestry reports handed to people with Southeast Asian, Pacific Islander, or Indigenous American heritage are almost certainly going to look meaningfully different from the ones available today, carrying more layers, more resolution, and a far more accurate picture of exactly how deep these lineages actually run. What won't change is the underlying reality that's already sitting in the genome, whether or not the technology has fully caught up to reading it yet. The broad nose someone inherited from a parent, a grandparent, or an ancestor whose name was never recorded anywhere carries a story that already exists, written into DNA tens of thousands of years before anyone had the tools to read it. That story survived an ice age. It survived the flooding of an entire continent. It survived migration across a land bridge into a hemisphere nobody from that ancestral population had ever seen before. And it survived quietly in the DNA of people who spent generations being told that the very feature carrying all of that history was something to fix rather than something to understand. We are only now, slowly and imperfectly, learning how to read it, and every year that passes, the story gets a little clearer, a little deeper, and a little harder to reduce to a simple label on a results page.